Hello everyone, my name is Cheng Gao, and today I'm going to present a genome-based influenza risk assessment using tree-guided sparse learning. Influenza A viruses cause large losses of human life and present a great public health challenge. Resorted viruses arise when genetic materials from two different viruses intermix. Genetic resortment between human and avian influenza A viruses facilitated emergence of all four low-end pandemic influenza A viruses. The human glutenide genes of 1918, 1957, and 1968 pandemic viruses are all of avian origin, and the PB2 and the PA genes of the 2009 pandemic influenza A virus are of avian origin. Risk assessment of potential resortments from contemporary endemic human influenza A viruses and zoonotic avian influenza A viruses is a key component in pandemic influenza preparedness. Conventional methods for assessing the emergence risk for novel influenza A virus often require laboratory creation of resortments and subsequent measurement of their ineffectivity, pathogenesis, and transmission in a mammalian system. In this study, we will focus on emergence risk, that is, the probability for novel influenza A virus to emerge. Because genetic resortment between human and avian influenza A viruses facilitated emergence of all four low-end pandemic influenza A viruses. Emergence risk for novel influenza A virus with gene segments from human and avian influenza A viruses, which is defined as resortment risk, has been the top priority in pandemic influenza preparedness, and thus will be the focus on this study. Given two influenza A viruses, each with 8 segments or 10 gene proteins, there will be 254 novel resortments. Specifically, resortment risk quantifies the probability of each of the 254 resortments to emerge when mixing a human influenza A virus and an avian influenza A virus. We aim to computationally identify two key features being associated resortment risk phenotypes, more specifically, a lower resortment replication efficiency in a mammalian system, synergistic combination of gene segments from 8 segments, and the synergistic combination of key features, for example, amino acid from a total of around 4,465 amino acids across the 10 gene proteins. This program can be naturally formulated as a sparse feature selection program because the prior studies suggested that a single or multiple mutation affect the viral ineffectivity, and our prior findings on influenza antigenicity suggested that the effectiveness of sparse learning on such biological challenges. The advantages of sparse learning over other conventional machine learning approaches are that its efficiency and generalizability generate accurate models using a small number of non-zero elements. A lower tree-guided machine learning model was developed and applied to identify both segment and residue network features associated with resortment risks using both segment and residue network features. The tree-guided model was divided by assigning each of the 8 segments or 10 proteins as leaves in the tree, with 5 internal nodes representing HA gene, NA gene, RNP complex, NS gene, and M gene. This novel tree-guided model was compared with L1, the lasso, L2, the ridge, L1, L infinite norm, sparse group lasso methods, and the tenfold cross validation showed that the tree-guided sparse learning model outperformed other models we compared. We found that RNP complex with non-zero weights were learned from the tree-guided sparse learning model, and this indicates that RNP complex will play an important role in virus growth as well as nulciferase activity. And a set of residues across RNP complex, HA and NA and NS1 gene were identified, and the majority of these residues are located with those functional domains reported from this from these domains, such as the contact region among RNP proteins and the effector domain of NS1 proteins. The computational model and the features we learn from this model can help with understanding the fundamental mechanisms for genetic resortment and with assessing emergence risk of influenza virus. Future study will focus on dealing with missing and undetectable values in the data set. Thank you so much.